All right, welcome back everybody. So today I thought we could talk a little bit about this web application for spatial transcriptomics analyses called spatialGE. And what spatialGE is it's a web interface that will allow users to analyze their spatial transcriptomic data without needing to program. So it's super helpful for those like biologists or basically anybody who is not trained in R programming or Python, those types of programming environments are able to use spatialGE to analyze their data. So here on the screen, I have spatialGE open, and we can kind of see the layout here. If we go to how to get started, there's some good documentation about how you can upload your data. If you get it directly from I don't know, you get it from 10x or you get it from NanoString, it'll be in a format to where you're able to upload it pretty easily to spatialGE. And as I had said, we don't need any programming for this, but if you do prefer programming, there's the spatialGE R package. And if we head over to the GitHub page, if you wanted to install this package, what you're able to do is do DevTools install and then FriendlyLab spatialGE, just like we had done with the in situ type for phenotyping our CosMx SMI data with DevTools install GitHub. We're able to do that and install this spatialGE package. If we scroll down to the bottom of the spatialGE GitHub repository, there's a link here that's friendlylab.github.io that is created with package down. And if we head on over here, we can see that there's this vignette about how to use the spatialGE package. We can go to spatially informed differential gene expression, detection of gene sets, so on and so forth. Oscar Espina, so if we go to the home page here, Oscar Espina has done a lot of work on making this as complete as possible. So there's vignettes on how to use it. There's really good documentation on everything. If we head on over to reference, we can see all of the functions that are written for spatialGE. We can look at stclust or spatial transcriptomics clustering, and we can see he's got really good examples, all the parameters and stuff that are used, so on and so forth. But we'll be using the web application today. The files that we're going to need to upload, we're going to use publicly available data. So if we head on over to the 10x genomics, I'll put a link to the datasets page down in the description. But what we're going to search for is mouse brain sagittal section. We're going to get a couple examples, samples here. But what we're going to use is the original version 1. I scrolled over. Version 1 here. And there is a sagittal anterior and a sagittal posterior. And what I have done is downloaded the sagittal anterior samples. So if we download or head on over, after clicking those, it'll have you sign up. You can use dummy email and whatever you want to do. But we can head on over to the downloads page, and the two files that we're going to need is this feature barcode matrix HDF5, as well as this spatial imaging data. And those are the two files that are required by spatialGE's web application. If we go into the folder where those are downloaded, I've kind of jumped ahead on the steps. We have these. We have a GZ for the barcode matrix, and then we have these, we have the H5s for the barcode matrix, and then we have the spatial tar GZ for the spatial imaging data, this guy here. But what we need to do is we need to extract first the GZs into our folder, and by doing that we'll get the tar. And I like doing it this way versus going into the tar and then extracting this, because now we have spatial, and since we have two files, we'll actually have two spatial folders. So if we just do the tar, what we're able to do is right click and then do 7-zip if you have that installed, and extract to the folder. So we have these two folders here. We have sample 2 and sample 1. Awesome. So let's head back on over. So sample 1 and sample 2, the pages on 10x look identical. So it's this guy and this guy, those 2 links. If we head back on over to our spatialge.moffitt.org, that's the web interface for the spatialGE package. We can sign in. So we don't have any projects yet. You'll have to sign up, and when you sign up, you're going to get an email that will have you click a link, and by doing that, then you are kind of activating your account, so to speak. But you can't just sign up and then start using it right away. You have to sign up, wait for the email, and then click the verification link. But what we can do is start a new project. We have our Visium data. Otherwise, we can upload generic data if we just have matrices. But we'll have Visium data, and then we'll call this one mouse sagittal. I want to make sure that I spell sagittal correctly. I gotta go back here. Aha, nailed it. Brain. Description, we'll just call it brain from mouse sections. Visium. And then we can go ahead and create. So now we have to upload our samples. So the first one we'll do is just the unnamed, unnumbered one. So that's going to be our section 1, and we'll call that one here section 1. So for the gene expression data, that's our H5 that we had downloaded. If we go back over here, we can drag in our H5. Perfect. Now we have to go into our spatial file folder of our first sample, and then there are 2 images here. And what we want to do is we want to use the high resolution image. For the tissue positions, we can also use, so we'll do the tissue positions, and then we want to upload the high resolution image. Let's see. Let's change our view here. Details. That looks like the high res PNG is going to be the one that we want to go with. So we will upload our high res, and then we have scaling factors as well. So we'll upload our scaling factors to there. So we'll import our sample and I'll upload it there. And then we'll add another sample. And this one is our section 2. So we'll go section 2, and then we have to get back to our folder. Add in our section 2. We'll do the last 3 files first. So you have our JSON, we have our high res image. Well, that's how the high res image looks. Pretty cool. Now let's go back. Got our high res image, and then we have our tissue coordinates. You can see that is required, the first two of them there. And then we'll upload our section 2 H5 matrix. And there we go. So now we have our 2 samples uploaded to spatialGE. We can add metadata since these are publicly available samples. We don't really have any metadata to go along with them. I'm sure there is metadata in here, but since this is just example data, we're not going to do that, but we'll import data. And here's what I was talking about before, is we can send an email as soon as we're done importing our data into spatialGE, which is super nice. And then we don't have to keep the window open since our samples are already up there. Okay, so now we have our data kind of uploaded. What we're able to do is do some QC and data transformations. You can see here on the left-hand side that we have our project, and then we have a bunch of these different tabs, not so much tabs, but things that we can do with our data. So we imported our data. Now that our importing is done, we can go on to the next one, which is going to be our QC and data transformation. We can see our two samples. We can see the different spots that have cells, the genes that were identified for those, so on and so forth all the way over. Let's go to filter. We can set some thresholds. So we want to keep spots or cells that have so many gene counts. Remember, if there is a spot that has 1 or 2 genes identified for Visium, you can go back to the original spatial transcriptomics video that I did to kind of learn about what Visium is. But it's a circle that encompasses many cells. So if you only have a few transcripts that are being identified, you probably have either an empty spot that has false, or it might be contamination, something that you don't want. But the counts here, we can drag this up. Let's just keep it low. Let's go 500. And then we'll keep spots with so many genes expressed, right? So you don't want a spot that only has 1 or 2 genes, but maybe really high counts. That doesn't make any sense since there's multiple cells there. So we can bump this up. We want maybe 500 genes in a spot being identified out of the whole transcriptome. Yeah, let's apply filters. And there we go. Now we have our samples filtered. We can go on to normalized data. We have log transformation, log normalization, or we can use SC transform. SC transform does take a little bit to do. It's not as easy as just taking the log of a value. So we'll stick with log and let's normalize. There we go. Now we have our transformed normalized data. We can see some density plots. There is a lot that have low counts. You can see normalized expression after transformation. There's still a bunch of zeros. So it's single cell or approaching single cell, where you have a few cells, you're gonna have a lot of zeros. But we can see that we pulled up some of our data. You can now see it's a little bit more spread out than here. It's not so focused at very low numbers as we see over here. Let's see, gene empty. SERP option. We'll stick with spectral. All right. It doesn't look like any genes are found. Let's make sure everything works. Let's make a quilt plot. Sunset variable. We'll do total counts. We'll do the first sample and the second sample. So we'll be able to see the distributions here between the two of them. Since they are sequential, I'm assuming they're sequential sections, hoping pretty similar. Yeah, they look pretty good. We can see some section here, some structure, excuse me. Here we have some interesting low expression or low counts here in the middle. With low counts in spatial transcriptomics, you have something like this, where there's multiple cells. You're not focused on cells specifically. There might be more connective tissue, for example, where you're taking up space, but there's not going to be any cell associated with that location. So like fibroblasts are going to create extracellular matrix where there's tissue, but there's not going to be any genes that can be identified there. Here, I'm guessing this is probably more structure based. We have high expression here, some high expression down there with more genes being counted. You can see our normalized data. It looks like there were some dropouts, spots where there was nothing. But yeah, so that is a quick introduction. And now we can see that we did our normalization and transformation. We have all of this opened up here on the left-hand side. If we want to go to visualizations, we can generate some plots. But yeah, so if you have questions, you can ask them in the description down below. Alternatively, he did make a really nice video here. His videos take a lot more effort than mine, which I give him mad props for. He made a really nice video about the introduction to spatialGE using the web application. So I'll put a link to this down in the description too. Head on over there. If you have questions, feel free to ask him. He is the expert in spatialGE. I just help. So yeah, let me know what you guys think.